microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells (RBCs), enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids and phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tail towards the inner part. This ensures that nonpolar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to phospholipids, membrane also contains cholesterol. Later, biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the cell membrane of the erythrocyte has approximately 52% protein and 40% lipids. Depending on ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of protein while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson 1972, widely accepted as fluid mosaic model, figure 8.4. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example, sodium potassium pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner, towards membrane side of the cell. The middle lamellae is layer mainly of calcium pectate which folds or glues the different neighboring cells together. The cell wall and middle lamellae may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum ER, Golgi complex, lysosomes, and vacuoles. Since the functions of the mitochondria, chloroplasts, and peroxisomes are not coordinated with the above components, These are not considered as part of the endomembrane system. Endoplasmic reticulum ER. Electron microscope studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum. Hence, ER divides the intracellular space into two distinct compartments, that is, luminal inside ER and extraluminal cytoplasmic compartments. The ER often shows ribosomes attached to the outer surface. The endoplasmic reticulum bearing ribosomes on the surface is called rough endoplasmic reticulum. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum. Rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. Golgi apparatus. Camillo Golgi 1898 first observed densely stained reticular structures near the nucleus. These were named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1 micrometer in diameter. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the ER fuse with cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its transphase. Golgi apparatus is the important site of information of glycoproteins and glycolipids. Lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrates, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Vacuoles. The vacuole is the membrane bound space found in the cytoplasm. It contains water, sap excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradients into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. The amoeba, the contractile vacuole, is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size, also considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter of zero point 0.2 to zero point 0.1 zero point 0.2 to 1 micrometer, average zero point 0.5 micrometer, and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is, the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae, singular crista, towards the matrix. Figure 8.7. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence, they are called powerhouses of the cell. The matrix also possesses singular single circular DNA molecule, a few RNA molecules, ribosomes, 70S, and the components required for the synthesis of proteins. The mitochondria divide by fission. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplasts contain chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophylls, and other are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastids of varied shapes and sizes with stored nutrients. Myeloplasts store carbohydrates, starch, example, elioplasts store oils and fats, whereas the eleuroplasts store proteins. Majority of the chloroplasts of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. The number of organized flattened membranous sacs called the thylakoids are present in the stroma, figure 8.8. Thylakoids are arranged in stacks like the piles of coins called grana, singular granum, or intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids enclose a space called lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in thylakoids. The ribosomes of the chloroplast are smaller, 70S, and the cytoplasmic ribosomes 80S. Ribosomes. Ribosomes are the granular structures first observed in the electron microscope as dense particles by George Pallet 1953. They are composed of ribonucleic acid and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunits. Figure 8.9. The two subunits of 80S ribosomes are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here S stands for Svedberg's unit for the sedimentation coefficient. It is indirectly a measure of density in size. Both 70S and 80S ribosomes are composed of two subunits. Cytoskeleton. An elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as cytoskeleton. These cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia, singular cilium, and flagella, singular flagellum, are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscope study of a cilium or the flagellum show that they are covered with plasma membrane. Their core, called exoneme, possess a number of microtubules running parallel to the long axis. The exoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of exonemal microtubules is referred to as 9.2 array, figure 8.10. The central tubules are connected by the bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from the centriole-like structure called the basal bodies. Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by the radial spokes made of protein. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cells. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the black basic dyes was given the name chromatin by Fleming. The interface nucleus of a cell when it is not dividing, has highly extended and elaborate nuclear protein fibers called chromatin. Nuclear matrix and one or more spherical bodies called nucleolus is singular. Electron microscope has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, example erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? The nuclear matrix of the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nuclei are present in cells actively carrying out protein synthesis. You may recall that the interface nucleus has loose and indistinct network of nuclear protein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46, 23 pairs chromosomes. You will study the details of DNA packaging in the form of a chromosome in class 12th. Every chromosome visible only in dividing cells essentially has a primary construction or the centromere on the sides of which Disc-shaped structures called kinetochores are present. Figure 8.12. Centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosomes can be classified into four types. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome and resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric centromere chromosome has terminal centromere. Sometimes a few chromosomes have non-staining secondary constructions at a constant location. This gives the appearance of a small fragment called the satellite. Microbodies. Many membrane bound minute vesicles called microbodies that contain various enzymes are present in both plants and animal cells. Summary. All organisms are made of cells of aggregate of cells or aggregate of cells. Cells vary in their shape, size, and activities functions based on the presence or absence of a membrane bound nucleus and other organelles. Cells and hence organisms can be named as eukaryotic or prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Cell Plant cells have cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates transport of several molecules. The endomembrane system includes Golgi complex, lysosomes, and vacuoles. All the cell organelles perform different but specific functions. Centrosome and centriole form the basal body of cilia and flagella that facilitate locomotion. In animal cells, centrioles also form spindle apparatus during cell division. Nucleus contains nucleoli and chromatin network. It not only controls the activities of organelles but also plays a major role in heredity. Endoplasmic reticulum contains tubules or cisternae. There are two types, rough and smooth. Helps in the transport of substances, synthesis of proteins, lipoproteins, and glycogen. The Golgi body is a membranous organelle composed of flattened sacs. The secretions of cells are packed in them and transported from the cell. Lysosomes are single membrane structures containing enzymes for digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm or are associated with Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate. They are bound by double membrane. The outer membrane is smooth and the inner one folds into several cristae. Plastids are pigment containing organelles found in plant cells only. In plant cells, chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is the site of light reactions and the stroma of dark reactions. The green colored plastids are chloroplasts which contain chlorophyll, whereas the other colored plastids are chromoplasts which may contain pigments like carotene and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, cell is the structural and functional unit of life.